board on this computer. The last thing that I ended on was starting the first half of protein synthesis, which again, protein synthesis occurs in 2 stages. So basically we have to take the information from DNA and we transcribe, so transcription, we transcribe that into a molecule of RNA. And then that gets taken and translated, so translation, to a protein. And what do we have on DNA? We have a sequence of nucleotides, specifically the bases, which are going to translate on RNA into something called a codon. I'm going to talk about that today. A codon is a triplet of bases. And then proteins, of course, are going to, oops, I was going to write protein again. Proteins are made of amino acids. So this right here is your, is what's called the genetic code. It's basically going from a nucleotide-based sequence. That's the important part, sequence. That's going to be transcribed into a sequence of codons. Again, I'm going to describe or explain what a codon is. Oh, there's Ellie. Do I call her Elle or Ellie?
I call her Ellie, but her real name is Elle.
I know I asked her before. Did I call you Elle or Ellie? Elle, Ellie.
Oh, Ellie, Elle.
Hi.
Hi. Maybe I'll just call her Ellie. Ellie. Here you go. I just made a new nickname, Ellie.
Ellie?
Why did I have a new one? That takes too long to say though.
Ellie.
I used to, a really good friend of mine used to teach physics at the school. And like one of the funniest thing that we did was we would take everyone's nicknames and based on your nickname, we would make you a new nickname. So for example, a very common nickname for Thai students is Best. So then Best automatically became Worst. So that would be their new nickname. It was so fun. I love doing that. I don't do that anymore because he's gone. So no.
Oh, that's sad.
Anyway, so back to biology. So the genetic code represents a relationship between the nucleotide-based sequence to the sequence of codons. Like I already said, I'm going to talk about what that is. And then the sequence of codons is going to determine the sequence of amino acids, which determines the function of the protein. So depending on which end you start from, the relationship between all of these molecules is the genetic code. In the next chapter, I'm going to start explaining how genes get expressed. Because remember, this all stems from DNA, sections of DNA, which is what we're transcribing and translating to make proteins. Those are genes. So you take a section of DNA, which is a gene, you're going to transcribe it into a molecule of RNA. That RNA goes to the nucleus and is translated into a protein. But the important thing to understand is that there is a very strong relationship. There's a direct relationship, not very strong. It's a direct relationship between the order of your bases, which determine the order of the codons, which determines the order of the amino acids, and ultimately the order of amino acids is what is going to determine what the protein is and what it does. So that's going to be the topic for today. Oops, I'm going to take that away. All right, so the amount and the types of proteins that are produced in a cell are going to determine the structure and the function of that cell. That's why the type of protein is so important, because you have over 200 different types of cells in your body. Each cell is going to have its own function. Each cell is going to produce different types of proteins and enzymes, and those proteins and enzymes are determined based on their amino acid sequence, which is determined by the sequence of codons, which is determined by the sequence of bases of DNA. That's what we're going to focus on, the relationship. How is all of this related? The most difficult thing in biology is learning all of these steps, like helicase unwinds the DNA double helix, and then RNA polymerase comes in and it transcribes the template of DNA into an RNA strand, and then but you have to remember to zoom out and look at the big picture. Like, what does this mean? How is this all related? Okay, so if, that's all going to come back, isn't it? Yep. All right, if different cells have different proteins, how do we determine the type of protein it's going to be? It's based on the amino acids, which types of amino acids and what order they go in. Remember, there's only 20 amino acids in the human body. Some of them we produce on our own. Some of them are what are called essential amino acids. These are ones that we cannot produce, that we're going to have to get through the food that we eat, which is a reason why it's really important to have a healthy, balanced diet so that you make sure you get all of those amino acids. So proteins are made of amino acids. The sequence of amino acids determines the protein shape, therefore the protein function. If you remember, probably not, all the way back to chapter 3, I believe, when we talked about the structure of proteins. Proteins work based on a physical shape and how that shape matches up to different parts of the cell. So again, genetic code. During protein synthesis, the sequence of nucleotides in mRNA are translated into a sequence of amino acids. So the genetic code is the relationship between the nucleotides and the amino acid sequence. Now, I have a graphic on the next slide, which is going to probably freak you out and be like, oh my God, Pang's going to start crying because she's going to think, oh my God, do I have to memorize this? No, girl. It is a map that is going to show you the genetic code. Okay, you ready? Boom, there it is. Okay, so keep calm and breathe. Basically, what this means is that we are starting, remember that RNA has 4 nucleotides. We have adenine, which pairs with uracil, so A and U. We have cytosine, which pairs with guanine, C and G. In the this is the RNA sequence. In the DNA sequence, we have adenine that pairs with thymine, and cytosine pairs with guanine. I didn't spell that right, but never mind. So we have A pairs with T, C pairs with G. When we transcribe DNA into RNA, when we see adenine, we're going to add a complementary uracil. When we see thymine, it's going to be adenine. And then, of course, these two pair with each other, right? So just wanted to make that clear. How this map works is, remember when I said that the, what do you call it? The nucleotide sequence transcribes into something called a codon. A codon is a 3-letter sequence. So you start in the middle. Let's say your 3-letter sequence is GCA. Okay, so we have here G, C, and then A. So the sequence G, this is your first one, C, your second one, A, your third one. So GCA. This translates to the amino acid alanine. Okay, these are all abbreviations, by the way. This little ALA is alanine. Let's say it's AAA, AAA. Okay, so we got A, and then the last one is A. That translates to the amino acid lysine. CAU, CAU, histine, I think, or not histamine, because that's cytokine. I think it's histine, something that starts with a his. Anyway, so that's a different amino acid. This is the genetic code. How these nucleotide sequences, the order of these guys, how they translate to the amino acids around the outside. Now, I want to draw your attention to 4 specific sequences. So we've got these 2, this one, and then, oh, it's under this one. Okay, so there's 4 sequences. There's only 4 sequences you have to remember. There's one, which is AUG. So we've got A, U, and then G, AUG. That translates to an amino acid called methionine, but you see also on the bottom it says start, right? because there has to be a signal for all of the RNAs, the mRNA, the tRNA, and the rRNA. There has to be a signal that tells all of these RNAs when to start assembling the protein. That is something called the start codon. And then you have 3 different codons that represent stop codons. Now you notice that it just says stop, 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 whereas down here it says met and then start, because there is an actual amino acid called methionine, which matches the sequence AUG. Oops, okay. Don't look at the screen, look at the iPad. A, U, and G represents methionine, but it also marks the beginning of every single protein. If a protein in the end, the sequence doesn't actually start with methionine, it'll be removed at the end, but methionine marks the beginning of every protein. Now, 3 codes that tell the RNAs to stop assembling the protein: U, A, U, A, G, and U, G, A. And I'm going to go over all of this again. I'm just trying to get you to understand this map and how this works. So if the end of the mRNA, because remember this is all on mRNA. So let's say you have A, U, G, C, C, A, G, U, 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 G, A. Okay. Let's say that's your mRNA sequence. You're going to break it down into triplets. Each triplet represents something called a codon. So you start with AUG, okay? And you go to your map and you say, okay, AUG is the start codon. So your tRNA brings methionine here. And then you look at the next sequence. What amino acid does that represent? CCA. So go back to your map. Use yellow. CCA represents pro. I think that's proline probably. So you put a proline here and you attach them together with a peptide bond, which is the bond that holds amino acids together. Next one, GUU. So go back to your map. I'll use green. GUU. So I start with G, U, U. That's valine. I can't remember. I can't believe I can remember all these. So you attach valine there. And again, you put a peptide bond. And then you get to UGA. UGA. Oh, that's a stop codon. So we're finished making our protein. In this example, my protein is exactly 3 amino acids long, which doesn't exist because there are thousands of amino acids long, but you get the picture. Again, I just kind of went through broad strokes describing how this works. Now we're going to go through and look at it more specifically. So again, there is a relationship between amino acid sequence. Sorry, I keep using that word. There's a relationship between the nucleotide sequence and amino acid sequence, and it is in a triplet of nucleotides on mRNA called a codon. So every triplet represents 1 amino acid, 1 codon, 1 amino acid. Codon is a triplet. Okay, really important. So it's important to understand that if you change this one into a C, you've now completely changed the amino acid. This is where mutations come in, and I'll talk about that, I think, in chapter 11 or 12. On my die. I don't know. Coming soon. How about that? A few codons don't code for amino acids at all. They signal for the translation of mRNA to protein to start and stop. So you have the start codon, AUG. Again, this is one of 4 you have to remember. You don't have to remember any of the other ones, but 4 codons you have to remember. AUG also happens to correspond to the amino acid methionine, which may stay at the beginning or it may be removed at the end. It depends on the protein you're making. But this is the signal for rRNA and tRNA. This is where the protein starts, okay? The start codon. The other 3 do not code for I'm using the wrong words, that's bad. They don't code for any amino acids, they signal for the rRNAs and the tRNAs to stop. Okay, it marks the end of the protein chain. That is UAA, UAG, UGA. I don't know if there is a mnemonic device or some kind of acronym that you can use to remember those. There are only 4 of them, so I don't think they're that hard. Okay, so last week I described transcription, which is transcribing the information from DNA onto a molecule of RNA. Today is translation. We're literally translating the sequence of nucleotide bases into a sequence of amino acid, which is what a protein is. Translation begins when mRNA leaves the nucleus and migrates to a ribosome or rRNA in the cytosol or cytoplasm. Every protein is made of 1 or more polypeptides. As I said, there are thousands of different proteins in your body. I know hemoglobin, for example, is made of 4 polypeptides. There's like one chain is here, and then there's another chain that's there, another chain that's there, and the 4th chain that's there. 4 polypeptide chains for the protein hemoglobin. But all of them are made of amino acids that are linked by peptide bonds. This is a specific name for bonds between amino acids. Pep means protein. There are 20 amino acids found in every single protein of every single living thing on the planet, which is pretty amazing because if you think about how many different forms of life there are, all of those living things use different proteins and enzymes, all made with only 20 amino acids. That's amazing. Each peptide chain can have hundreds or even thousands of the 20 amino acids. The key, the absolute most important, vital, essential key is the sequence of those amino acids. If you change the sequence of the amino acids, you change the way the protein folds into a particular shape, you change the function. And the sequence of those amino acids, sorry, my dog's wandering around the room in the background. The sequence of the amino acids is completely dependent on the sequence of nucleotide bases on mRNA. This is an example of 2 polypeptide chains where I've changed one of the amino acids. So here you have 2 polar amino acids. Because they're both polar, they will form a hydrogen bond and form a fold in the chain at this point. If I change the 3rd amino acid to a non-polar amino acid, you won't have that hydrogen bond formed, and you see it completely changes the shape of the protein. If you change the shape of the protein, you change the function. This is an example of the 4 levels of protein structure. You've got primary structure, which is just the chain of amino acids, the sequence of them. The secondary structure is either something called the alpha helix or the pleated sheet. This is basically just the folding like I showed you here. This is how it folds like this. Tertiary structure is the overall shape that the polypeptide takes when it's folded. And quaternary structure is if you have more than one, what do you call it? More than one polypeptide chain. So like I said, hemoglobin is 4 polypeptide chains. So you see the 1, 2, 3, 4 chains here. So I can't even read. These are just different proteins, just to show you the different shapes of them, right? All of these shapes occur between the secondary and tertiary structure. So you see how important shape is? And shape is purely due to sequence of amino acids. Okay, I just said that. I said that too. All right, so let's have a look at translation. Just to point out our 3 structures here, we've got mRNA here, which is that single strand. This contains the code for the order of amino acids. This blobby thing, which I like to call my ribosomal hamburger, is the rRNA. You've got the large subunit on the top and the small subunit on the bottom. And this little T-shaped clover-shaped thing is the tRNA. And you can see the tRNA here, we start with methionine because we've got AUG. See it right there? AUG. Let's see if I can draw AUG. That is the start codon. And so that's where methionine is going to sit. And you can see amino acid methionine right here. And then what happens is this entire ribosome is going to shift down the mRNA molecule by 1 codon unit. So I kind of covered it up. There we go. Let me try to draw it better. Ooh, so we can see all of the codons. It's going to shift 1 unit down, and then it's going to sit on top of the next codon, which is UGU. And UGU, you're going to see the tRNA bring in the amino acid that matches UGU to form the next amino acid in the chain like that. So step 1 obviously is putting all 3 RNAs together. You've got rRNA, tRNA, mRNA. Right. Now, if we zoom in on tRNA, how does tRNA know which amino acid to bring? It's not really about how tRNA knows which amino acid to bring. It's about which tRNA molecule matches the codon. So for every single codon you have a different tRNA molecule, because tRNA has on the bottom something called the anticodon. The anticodon is just complementary to the codon. And this is how you know. It's not like you have one type of tRNA and it has to look at the codon and translate the codon and say, okay, what amino acid does that match? No, no, no. It's that you have a tRNA molecule that is complementary to every single codon. So if you have, I don't even know how many if I go all the way back to, we got issues. Hold on a second. If we go back to here, if we go back to the genetic code, I'm sure there's some kind of math thing that you can do to figure out how many different combinations of these letters there are. I'm not a mathematician, I don't know, but it's a lot, right? It's a lot. So for every single combination you have here, there is a matching tRNA molecule that knows, because of the anticodon, it knows which amino acid to bring. So the amino acid is going to attach up here at the 3' end, it's the amino acid attachment site. And so you've got the codon, GAG, whatever amino acid matches GAG, this tRNA molecule is the only one that fits because C pairs with G, U pairs with A, C pairs with G. So this is the tRNA molecule that matches GAG. And so there's a specific amino acid that will fit there. That's how it knows. It's all about that complementary base pairing. So the codon corresponds to an amino acid and the anticodon corresponds to a codon. Okay? The amino acid corresponds to the codon and the anticodon matches the codon. So this, how do I do it? Like this amino acid is because of that, GAG, and GAG is complementary to CUC. And so that's how the relationship works. All right, the tRNA carrying methionine, which is the same as the start codon AUG. So if my start codon is AUG, the anticodon, the complementary, A pairs with U, U pairs with A, G pairs with C. So the tRNA that contains the anticodon UAC will bring methionine. It can be removed later if the final protein or enzyme doesn't start with methionine. So here's what I mentioned the other day about 3 sites. There's the E site, which is the exit site. There's the P site, which is the peptidyl site, and the A site, which is the active site. I don't think you have to remember all 3, but if you continue studying biology in university, they will go over those again. The tRNA that carries the correct amino acid pairs its anticodon with the codon on mRNA. Again, it's always based on the base pairing rules. The ribosome is going to detach methionine from the first tRNA, and it's going to form a peptide bond between the first and the second one. So the first tRNA leaves, that's here. That's why it's called the E site, the exit site. The ribosome, the whole thing, is going to shift down one codon. So this is the P site, this is the A site, this is the peptidyl. Peptidyl, is it YL? I can't remember. Anywhere, that's where the peptide bond forms. That's why it's called the P site. The A stands for active. That's where the new tRNA comes in. And the E site, exit. So there's a free tRNA that has released its amino acid, and it's free to leave and go get another one if there's a sequence later on that corresponds to the same amino acid. There you go. So when we move this whole thing, I wish that I could actually select it and move it down one. When we move it down one, this is going to be the E site, this is going to be the new P site, and then the next one will be the new A site, and you keep moving down. I do have video, so don't worry about it. As the mRNA, as the ribosome moves down the mRNA, you're going to keep adding. You can see our protein chain is getting bigger and bigger as you move down the mRNA molecule. Eventually, you're going to reach one of the 3 stop codons, UAA, UAG, or UGA. One of those 3 signals everything to stop. The polypeptide chain will be released, which you can see here, and the tRNA and the rRNA are also released. Here we go.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has 2 binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid.
Hold on, I know he said 2, but I just want to point out that this is the exit site here.
Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino...
Oh, why do you go so fast? So again, you want to make sure that the anticodon is complementary to the codon so that the tRNA brings the correct amino acid. So if the codon is UUU, the anticodon is AAA, and this specific tRNA will bring that amino acid.
The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
Okay, and everything disassembles.
Now we
Okay, so again, this I've showed you the transcription of this one before. This is all a computer model based on the actual structure of the molecules. So we're starting with the mRNA just coming out of the nucleus. These are the nuclear pores here. You can even see the tRNAs here in the background. There's the anticodon up there.
the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, the mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript.
This is happening inside a ribosome, by the way. They've zoomed in a lot. So mRNA is here at the top, and you can see that it's going codon. So 1, 2, 3 codon, and anticodon is complementary.
These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed.
Here's the protein coming out. Okay, so once the stop codon is reached, all of the components of translation come apart. So the polypeptide chain is released, the mRNA, the rRNA, and the tRNA. Several ribosomes can translate the same mRNA transcript at the same time. This is because a new ribosome starts translating mRNA almost as soon as the previous ribosome moves aside. So you can have basically like 2 or 3 or 4 or a dozen ribosomes like back to back to back if you need a lot of the same type of protein. Because prokaryotes do not have a nucleus, translation can begin before the entire mRNA molecule is even finished being transcribed. Because prokaryotes don't have a nucleus, transcription and translation occur in the cytoplasm. So even as you're making mRNA and it's being made, you can start translating it right away. In eukaryotes, we have a bit more control over the process because transcription and translation occur in different locations. Transcription occurs in the nucleus, translation in the cytoplasm. This gives our cells time to adjust or even manipulate the mRNA before it leaves the nucleus. Okay, I don't know how much is left, but I am spent. Oh, there's only 2 slides. Ugh, okay. I'm just going to finish it then. Right, so you might have or might have not heard of something called the Human Genome Project. A genome is an organism's entire set of DNA or nucleotide bases. So basically from start to finish, the entire sequence of nucleotides in a DNA molecule of a specific organism. That's called, excuse me, a genome. Over the course of 10 years, I think, was from like 2000 to 2010, there were many organizations in many countries that were working together to map out what's called the human genome. They deciphered the order of 3.2 billion base pairs in our 23 chromosomes. The human genome, I love this statistic. The human genome is so large that if you tried to read it aloud, just like AACAGGCTTAACC, whatever, it would take you 10 years to read out our genome. That is how huge DNA is. The challenge now becomes to learn what those sequences actually mean. And oof, I forgot to bring it. I went to school on Monday to get some materials, and I meant to pick up, I've got a map. Oh, I can just download it again. I've got a map of the discovery since the Human Genome Project. We've been able to identify genes on specific chromosomes. So for example, certain types of cancer, we know where on which chromosome that they occur. So if we're able to associate a specific sequence of nucleotides that we've been able to map out with the location of a gene that is associated with cancer, especially now with a new technology called CRISPR. I can talk about that tomorrow. We might be able to edit, like literally edit DNA so that we can try to prevent or even cure things like cancer. There is a new field. Oh, it's too bad Pete's not here, because is Pete here? I don't know. I don't know, because there's only 8 people and I can't remember if he came in or not. When I had you do the career assessment thing, bioinformatics was the first thing that came up for Pete. This is a new field. I don't know, relatively new, maybe like 5 or 10 years. And for science, I guess that's getting pretty old, but a growing field called bioinformatics is literally combining computer science with biology. So you're basically collecting data in biology like DNA sequences, and you're using computers to analyze that data. It's really, really, really cool. So we can program computers to help interpret a lot of DNA sequences, and we can predict where genes are located. And we have so far been able to identify about 30,000 genes in the human genome. Understanding which of these genes control which functions can help us diagnose, treat, and prevent genetic disorders, cancer, and other diseases. So it's a pretty exciting time in science if we could just get people to